Clinical trials confirm that the renal assist device, or the RAD as it has become to be known, could safely treat acute kidney failure in critical care settings. However, it's relatively large and requires a lot of utilities. So its adoption for routine treatment of dialysis patients is going to be limited by the need for labor-intensive operation, the huge size, and the cost associated with running it. So we decided to miniaturize the RAD technology to become eventually what will be the basis for the implantable bioartificial kidney. To do that, we leveraged semiconductor nanofabrication technology that is available to create computer chips, that's available to create, smartphones, and use that to miniaturize the membranes for filtration. These high efficiency silicon membranes allow for operation within the body without the need for a pump. They are coated with biocompatible coatings that do not elicit coagulation of thrombi or blood clots. And they also stabilize the kidney cells inside the device without triggering an immune reaction. And this combined altogether results in a compact, efficient, and implantable device. Yeah, so the RADS clinical trials highlighted two major challenges: the size and the pump requirements of traditional dialysis infrastructure, mainly the machines and the space and the utilities that are required, as well as the large volumes of water needed for dialysis. While we're inspired by the success of the standard hollow fiber polymer membranes that's used in dialysis, we recognize that they are very different from the native kidney's tissue, which are highly elongated, slit-shaped filters and with uniform pore size, very different than what the hollow fiber membranes are. So by adapting the semiconductor nanofabrication technology, we're able to produce new membranes, which we call silicon nanopore membranes, with kidney-like slit-shaped pores and designed with a geometry that's tailored for implantation. By making these membranes look more like the kidney, but in a much more compact format, and coating them with the right chemistry to make them blood compatible, we have created a device that allows for precise filtration, can be implanted to the blood vessels and operate continuously without any adverse reaction. And that is what forms the core of our artificial kidneys design. Yeah, so to get to the clinical trials, especially the first in human studies, we need to overcome two additional steps. One is you have to complete the preclinical studies to demonstrate the safety of our device and its efficacy in models of kidney failure that are preclinical, meaning in animals. So we have to do those studies in an animal model so we can show that we can actually treat an animal that has kidney failure and keep them alive. Once we have that data, we can then combine that with the tests we must do about material safety and compile that into a packet for regulatory approval from the FDA. And these two steps must be done before we can go to the first human testing. So what are the challenges that could slow our progress? Fundamentally, it is going to be getting adequate funding to do the work. Once we have that, we actually have to successfully complete the preclinical studies so that they meet the bar of the regulatory agency. And for us, demonstrating that our device produces enough urine, or put another way, operates consistently over time. While these challenges are there to be overcome, they're fundamentally technology based. What I mean by that is we're not waiting for new science to be available for us to proceed. We have that necessary science. What we need is the funding and the resources and the organization to put everything together and go through these preclinical studies and then get regulatory approval. Our device will provide continuous ultrafiltration instead of the intermittent filtration that dialysis provides. Continuous ultrafiltration more closely mimics the natural kidney function by maintaining both the balance of fluids and electrolytes. This approach will reduce cardiovascular stress, improve blood pressure control, and minimize the fluctuations in waste product levels associated with conventional intermittent hemodialysis. Overall, this would lead to long-term outcomes that are better for the patient in terms of health, with fewer complications and improved daily well-being for the patient. The artificial kidney contains cells from an external source, but these cells are going to be isolated from the patient's immune system, and that's by design. The pores in our silicon membrane are big enough to allow nutrients, waste, and excess fluids to move between the bloodstream and the bladder through our device. But the pores in the membrane are small to keep out the immune components. This design feature, which we have engineered through the semiconductor nanofabrication technology, allows our device to function without triggering the symptoms of rejection. The bioartificial kidney will dramatically improve patient outcomes by providing continuous kidney-like filtration and reducing the complications associated with traditional dialysis. Patients will experience greater freedom, increased mobility, fewer hospital visits, and an enhanced quality of life overall. From a healthcare perspective, the widespread adoption of the bioartificial kidney will lower long-term costs for society, insurance companies, by decreasing hospitalization, reducing the infrastructure associated with dialysis, and extending patient lifetime while maintaining their health and independence. To summarize, we have shown the feasibility of the bioartificial kidney in early preclinical studies. We have constructed the filter unit of our device. We've combined that with the cell component of our device to create a functional bioartificial kidney that produces urine. It's at a prototype stage, but confirms this approach that's inspired by the Renal Assist device really works. And what we have to do now is to scale up the technology, both in size for human treatment and in duration, so that this will work for longer than what we've done in the lab, until we get the data that we can take to the FDA for human studies. And that's our focus right now.